VitalTherapies is a biotherapeutic company developing a cell-based system targeting the treatment of acute liver failure. The liver performs a wide variety of vital functions. The liver is the mainstay of protein metabolism, is involved in the processing of fats and sugars, and produces cholesterol, triglycerides, and bile. The four main areas of liver function are metabolic, blood filtration and detoxification, synthetic activities, and immune response. The primary liver cell, the hepatocyte, is thought to be responsible for approximately 500 or more specific biologic processes. Hepatocyte cells are responsible for the removal of toxins and the synthesis of proteins essential for life. In a healthy person, the liver has a remarkable capacity for recovery and regeneration, enabling the organ to combat the continuous damage to hepatocytes that result from their detoxifying function. In patients with viral hepatitis or chronic alcohol consumption, this regenerative function can be compromised and overall liver function may slowly deteriorate. In turn, this can lead to increased susceptibility to infection and risk of bleeding, which can induce acute exacerbation of the liver disease, a serious medical condition requiring treatment in the intensive care unit and often leading to liver transplantation or death. Another cause of acute exacerbation of liver disease is excessive consumption of alcohol, which can lead to a serious condition known as severe acute alcoholic hepatitis with a high mortality risk. Other causes of acute liver failure include acute toxicity, for example, overdose of acetaminophen and drug reactions. The stages of liver disease. Stage 1. Beginning of liver damage. The damage has started with some slight scarring. Stage 2. Moderate liver damage. The scarring advances in the liver and surrounding blood vessels. Stage 3. This stage sees significant liver damage. The liver becomes fibrotic or scarred and it connects with other scarred areas. Stage 4. Final stage of liver damage, also called cirrhosis. The liver is so severely damaged that it can no longer function properly. ELAD, a human cell-based bioartificial liver support system. ELAD is an allogeneic cellular therapy system under development, incorporating our human liver-derived cells known as VTL-C3A cells, combined with a reusable ancillary delivery system and single-use disposables. Our VTL-C3A cells have been shown in non-clinical studies to retain many of the specific metabolic processes and pathways of normal human hepatocytes, the primary functional cell of the liver. During ELAD treatment, blood is drawn from the patient via a central venous line and then passes through the tubing where plasma ultrafiltrate is generated from whole blood using the ultrafiltrate generator. Heparin, a widely available anticoagulant drug, may be administered during ELAD treatment in certain patients to prevent clotting in the whole blood circuit. The patient's plasma ultrafiltrate then passes into the 4 ELAD cartridges, which collectively contain approximately 440 grams of VTL-C3A cells. It passes through hollow fibers which allow appropriate two-way transfer of toxins, metabolites, and nutrients, mimicking liver function. The fibers, made from a semi-permeable membrane, permit passage of macromolecules from our VTL-C3A cells to the patient's plasma ultrafiltrate. At the same time, these fibers permit the passage of toxins such as bilirubin and nutrients such as glucose from the plasma to our VTL-C3A cells. The patient's ultrafiltrated plasma is treated by our VTL-C3A cells in a single session of continuous therapy lasting between 3 and 10 days. A centrifugal pump circulates the plasma ultrafiltrate at 2 liters per minute, allowing multiple passes through the cartridges before being returned to the patient's bloodline. Treated plasma ultrafiltrate is then sent through a cell filter. The treated ultrafiltrated plasma is recombined with the patient's blood, and then the blood is returned back to the patient via the central venous catheter. Meanwhile, the ancillary delivery system monitors temperature, pH, and oxygen concentrations in the plasma ultrafiltrate in order to maintain the VTL-C3A cell's viability. The ELAD system was designed with the proposed intent to supplement hepatic function. ELAD is currently in Phase 3 trials for subjects with liver failure. The primary objective of our Phase 3 trials is to evaluate the safety and efficacy of ELAD with respect to overall survival. For more information on our clinical trials, please visit our website or clinicaltrials.gov.